We got to meet Dr. Brunetti yesterday. He is an associate investigator in the molecular therapy program and a professor of medical genetics in the Department of Translational Medicine at the University of Naples, Federico II in Italy. His interest is understanding the pathogenetic mechanisms and developing novel treatments for genetic diseases. And he is actively working in the clinic and in the laboratory and has identified several novel genetic syndromes. His major research effort has been focused on investigation of gene therapy and small molecules for therapies of genetic diseases. His laboratory has been involved in preclinical studies to support the efficacy of small molecule-based therapies for maple syrup urine disease, pyruvate dehydrogenase deficiency, urea cycle disorders, and Myhre syndrome. As a clinical investigator, he is currently involved in liver-directed gene therapy clinical trials. His research activity has been recognized by awards of the American and European Societies of Gene and Cell Therapy. I want to welcome back Dr. Brunetti.
Thank you. Thank you again for the nice presentation, and thank you for having me here today as well. And I wanna try to tell you the story of how I got interested in this group of disorders that includes Myhre syndrome and some other defects of the TGF-beta. And really, the interest started from a clinical observation from a patient that I saw on the genetic consult at the time when I was a genetic fellow at Baylor College of Medicine. This is a patient I saw with my mentor and friend, Carlo Basino, back several years ago now. And this patient presented with the tracheal stenosis, short stature, brachydactyly, joint limitation, a coxavara that you can see from the X-rays. And they also had thickening of the mitral valve leaflets. And this was several years ago because the gene known for Myhre syndrome, for example, was known, and because of before also the gene for the condition that we suspected clinically was known. At around the same time, I also saw again with Carlos these two siblings that had much milder phenotype, had the J-shaped vesella, short stature, brachydactyly, and also a pulmonary valve stenosis. So there are some features in both conditions that led us to some suspicions of lysosomal storage disease, but all the testing came up to be negative. And then we ended with this clinical diagnosis, that is Genophysic Dysplasia, that has several of the features that are seen in those two patients, and also has similarities with the lysosomal storage diseases. This condition was known to have a characteristic happy face. From this feature came the name of a Genophysic, a short stature, high-pitched voice, glaucoma, cardiac valve involvement, short hands and feet, joint contractions, and thick skin. Again, this was a few years ago, and before the genes for Genophysic Dysplasia were identified. And you heard from Valerie earlier this morning about these different genes. Actually, Valerie found the genes for all of these different types of Genophysic Dysplasia that are now called type 1, 2, and 3, and they have different mode of inheritance. One is autosomal recessive, and is due to ADAMTSL2 mutations. And the other two are autosomal dominant, and they are due to fibrillin-1 or LTDP3 genes. What is common to all these genes is that, here are the, and in fact, we suspected this condition that we confirmed because our first patient was found to carry a heterozygous missense variant in the fibrillin-1 gene. And similarly, another missense mutation was found in the two siblings. So at least we were right that about the clinical diagnosis, because it was later confirmed by the genetic study, although the two cases are have some differences, but no, this is not surprising with many genetic diseases having a spectrum of various clinical manifestations. But what these Genophysic Dysplasia have in common, the three genes, is the involvement of the TGF-beta pathway. This is also, as we heard several times during this meeting, is also a feature of Myhre syndrome. And all these conditions are associated with an increased activity of the TGF-beta, which is an important signaling molecule. So here is, for example, you see a cell that is making and producing TGF-beta and uses different proteins that are involved in the secretion of the matrix as well as the TGF-beta. And then the TGF-beta is captured within the extracellular matrix, and can also signal through the TGF-beta receptor to neighboring cells, as shown here in this slide. But you have seen this type of slide several times in different formats throughout the meeting. And here are the genes for the Genophysic Dysplasia, type 1, ADAMTSL2, the type 2, the fibrillin-1, type 3, LTBP3, and here is our SMAD4, which is responsible for Myhre syndrome. Interestingly, fibrillin-1 is also the gene that is responsible for Marfan syndrome, which is another connective tissue disorder, but presenting in a very different way. Actually, Marfan syndrome presents in an almost opposite phenotype. While Genophysic and Myhre syndrome have short stature, Marfan syndrome has high stature. Short hands and feet are observed in Genophysic and Myhre syndrome, whereas long hands were typically known as arachnodactyly are seen in Marfan syndrome. The joints are contracted in Genophysic and Myhre syndrome, and they are hypermobile in Marfan. The skin is thick in the Genophysic and Myhre syndrome, and is soft in Marfan. But both conditions have an increase of TGF-beta. How this is possible, I think it's still a mystery. It's something very interesting that needs to be understood still. But what is also clear from these disorders and from the biology underlying this disease is that the extracellular matrix is not only working as a mechanical support for cells and tissues. It serves also, it clearly integrates cells into tissue, influences the cell shape, differentiation, and development, but also has important functions in signaling, and is a reservoir for signaling molecule, like in the case of TGF-beta. And therefore, if there are defects in the extracellular matrix, there will be secondarily defects in the signaling molecules. And this is the mechanism that is thought to be responsible for both Marfan, Genophysic, and Myhre syndrome, a defect in the structural proteins of the extracellular matrix that alter the TGF-beta availability. So this is what I showed in the talk I had yesterday, in which we found that Lozartan, that is an inhibitor of the TGF-beta pathway, can improve the extracellular matrix deposition in fibroblasts of Myhre syndrome. And obviously, the questions we asked was also to see whether the extracellular matrix deposition defect could also be improved in Genophysic dysplasia. We didn't have many fibroblasts from Genophysic dysplasia, at least not as many as we had about Myhre syndrome, but we could confirm in a cell line from a Genophysic dysplasia patients carrying a fibrillin mutation, the mutation we have seen in the two patients. When these cells are incubated with Lozartan, there is an improvement in the extracellular matrix deposition. Actually, if you compare the vehicle-treated cells with the Lozartan-treated cells, you can see a better deposition of the fibrillin-1 that is staining in red in this panel that is highlighted. So there is also something interesting that was several years pointed out by Spranger about genophysic dysplasia, that is the accumulation in the within the cells of inclusions that are storage-like inclusions, and in fact, genophysic dysplasia was initially thought to be a lysosomal storage disease, and it's not surprising then that we suspected our patients initially to have a lysosomal storage disorder. And I was also fascinated by the comment that Dr. Kinane made on the ribs in his patients that are looking thickened, as sometimes seen in lysosomal storage disease. So these inclusions were detected in chondrocytes and fibroblasts of patients with genophysic dysplasia. And we had the opportunity to also evaluate cell lines from another patient with the genophysic dysplasia type 1, that is the autosomal recessive form of genophysic dysplasia due to ADAMTSL2. And we could confirm that there is the presence of inclusions in the skin fibroblast. So here, S3 is a patient with the genophysic dysplasia, and here is a wild-type control. And you can see here the arrows in the S3 that are pointing towards these large inclusion bodies. We also looked at the presence of this inclusion by electron microscopy in other genophysic dysplasia patients, S1 and S3 here, and also in Marfan syndrome fibroblasts. And we confirmed the presence of these inclusions in all three cases. Interestingly, we also tried to see whether these inclusions are lysosomes, and in fact, here is an immunogold staining. So we are labeling with LAMP1 with the immunogold particles. Here in the wild type, you can see the blue area that is highlighting a lysosome in a wild type control cells. And in here, wild type control is another wild type control, number 2. And then we have cell line fibroblasts still from a genophysic dysplasia patients that that have abundant and several inclusions that are all surrounded by LAMP2, suggesting that those are of lysosomal origin. We also counted the number of lysosomes in the cells, and in patients with the genophysic dysplasia, we could see a significantly increased number of lysosomes. Also, we look at expression levels of different lysosomal genes again, this is mRNA levels, and those are increased in genophysic dysplasia fibroblast. So I guess our conclusions were that Lozartan can improve the extracellular matrix deposition also on fibroblasts carrying a fibrillin-1 mutation. We confirmed a relatively old observation made decades ago on skin fibroblasts from genophysic dysplasia patients that are showing intracellular inclusion of a known material, although this material is enclosed within lysosomes because they are LAMP1 positive. And these inclusions are also associated with the upregulation of lysosomal genes. This taken together with some of the features of the disease that are resembling the lysosomal storage disease is an intriguing finding that needs, in my opinion, further investigation. Also, the pathophysiology and the clinical relevance of these inclusions is totally unknown. And so the open questions, I think, is that there are changes in the extracellular matrix deposition in genophysic and Marfan syndrome fibroblasts that appear to be actually quite similar to Marfan. But how this similar observation made in a fibroblast and a similar increase in TGF-beta activity can result in opposite phenotypes remains unknown and remains a very fascinating question to be addressed. Whether the storage material is relevant is also something, it could be a simple artifacts of fibroblasts growing in culture, but we typically don't see that in the wild type fibroblast. But whether those are relevant for the disease pathogenesis is still unclear. With those open questions, I will want to finish by acknowledging the Pasquale Piccoli in my lab that did some of the studies in the cells that I presented, our electron microscopy facility by Roman Polishuk, and Carlos Pacino for the clinical insights into these cases. And I'm looking forward, as some of you may know, in a couple of hours there will be a big match, at least for Europeans. There will be the final of the European Championship, and hope Italy will do better at that game. And I will leave you for that game in the next hour or so. And with that, I thank you again for inviting me to participate to this very interesting first Marfan Syndrome Foundation meeting, and I will be happy to take questions.
Thank you, Dr. Brunetti. We have one question for you right now. What is your current greatest obstacle to advancing to the next steps of your Lozartan research?
Well, it goes back to the issue we discussed yesterday. We really need to do one of those studies that provide good evidence of efficacy. The ones that Mark in his talk mentioned would be the ideal study. So a randomized, controlled, double-blinded study. It's not impossible. Actually, it should be relatively straightforward to do because Lozartan is a relatively safe drug. We have been giving it to patients. So I don't see major obstacles. It's totally feasible. It requires efforts, of course, but we will try to do our best to move further with that.
Great. Yeah, I wanted to ask Dr. Lindsay if he'd like to join the conversation. He had a comment in the chat. Mark, are you there?
Yeah. I was just I was interested in the pictures of the inclusions. It's interesting. We've been looking at autophagy and dysregulated autophagy in Marfan cells, and we see a lot of these same structures which appear to be autophagosomes. And it's interesting that the cells seem to upregulate autophagy, but it's sort of a abortive process in that the autophagosomes aren't trafficking to the lysosomes, and maybe, at least in our hands, mediated by a deficiency of LAMP2 and several other autophagy-related proteins that don't get upregulated. So it's interesting you see those inclusions in the Myri cells as well. We haven't looked.
Yeah, well, if you talk with somebody that is knowledgeable about autophagy, it will probably, this is what I did. So the most of the time they say, of course, if you have the inclusions in the cells, you will have an autophagy activation. There's no question about it, because it's a normal reaction that cells have against something that, that is accumulating. And in fact, we did look at the markers of autophagy in those cells, and they are upregulated, but this is totally expected. Now, I guess what is leading to those inclusions, and is there any TGF-beta hyperactivity that is the cause of those autophagy dysfunction or accumulation of inclusions? I think those are interesting questions to be addressed.
Yeah, absolutely. very interesting and similar cellular phenotype with a reciprocal activation of TGF-beta. So, sorry, let me mute myself. Thank you for your answer.
Thanks. All right. Thank you so much, Dr. Brunetti. That's a great.
I think Angela is raising this hand, not the electronic hand of the.
So first of all, thank you, bravo, grazie. And I just want to thank the Myhre Syndrome Foundation again for letting us do this, because there has been such pleasure and chatter behind the scenes after each of us gives a talk, you know, congratulations, good talk. And the reason is that I think a talk really does gel. It coalesces, it forces one to review one's data that you've been sitting on for a couple of years, and you put it all together the night before, as did I, and now we get to hear this. So it's so enriching. Also because each of the speakers generally gives some reference point about how they got into Myhre syndrome, and that is also very helpful. So it's just been wonderful, and we thank you for letting us do this. So I had made a pitch that we need more tissues. Just pretend somebody had, I can't think of anything, some type of surgery, and there was an opportunity to have skin. In general, we try not to get called the morning before. In general, it's nice to know in advance. With other groups that I've worked with, people have said, well, that's a great idea, Dr. Lim, but where does it go? And you can't put the burden on the local hospital. So now you know everybody, you know, we know each other, and we could try to get in touch. And so at the very least, the local hospital could do basic histology, you know, just a basic review. You could ramp it up and ask for EM. But sort of what we'd really like to do is have it as a biologic sample to culture as well as look at. And so this is our first step, to make the pitch, and that's what it is. And I guess the other thing I wanted to add to Nicole is, and Mark, you know, here we are, we've been talking about pathways, and people understand they're interacting, they overlap. And I like to bring in stiff skin syndrome, and yet another role for fibrillin, and how I have become so interested in it now. And I think you two already have more experience. Want to chat about that?
Well, I think it's fascinating. It's something, there is clearly something going on there that goes, from stiff skin syndrome to Marfan to Myhre. Common pathway, but different phenotypes. I think somebody has to find out what's going on and why there are such opposite phenotype with diseases that are affecting the same pathway. But it's not going to be easy to do, but certainly it's a fascinating question that is open.